Thanks everyone for coming back for this afternoon session, and thanks especially for the organizers to giving me the opportunity to present some of our data today. So my name is Melanie Blokesch, and I have my lab at the Swiss Federal Institute of Technology in Lausanne, EPFL for short. So in my lab, we're interested in Vibrio cholerae. So here I'm showing a scanning EM of Vibrio cholerae, and I'm sure you're all familiar with the fact that this is the causative agent of cholera and an important pathogen. And it leads to very severe diarrheal disease, and that can be fatal if left untreated. So just to give you an impression, a human adult, if infected with Vibrio cholerae, can lose up to 20 liters of stool a day. And despite the fact that, of course, us in developed countries, we're not much in contact with Vibrio cholerae anymore, there's still around 4 million cases of cholera worldwide, of course, mostly in developing countries where there's a lack of hygiene and clean drinking water. So Vibrio cholerae actually is a marine bacterium, and it lives in the aquatic environment. And it can do so either as a planktonic free-swimming bacterium, as shown in this scheme here, but it's thought that more often, though, it associates with plankton, and that can be either zooplankton or phytoplankton, or phytoplankton and zooplankton is shown here. And based on studies, mostly by Rita Colwell and her collaborators, it's currently thought that the transmission to human in endemic areas might occur because Vibrio cholerae interacts with zooplankton, so tiny crustaceans. And that's also what my lab is interested in. We're interested in the association of Vibrio cholerae with zooplankton and crustacean. And what I'm showing you here is a scanning EM micrograph that we took of Vibrio cholerae colonizing a exoskeleton, a crustacean exoskeleton, and they form very thick biofilms on these surfaces, and that's something that we would like to study also in the future. And so I want to remind you from your Biology 101 class that actually the exoskeleton of crustacean is made of a polymer that's called chitin. And it's a polymer of N-acetylglucosamine as shown here. And it's extremely important because it's the most abundant polysaccharide in aquatic environments. Just what cellulose is on Earth, chitin is in the aquatic environment. And so a while back, while I was still a postdoc with Gary Skolnick at Stanford University, what we have actually shown that once Vibrio cholerae grows on these chitin surfaces, it induces a developmental program that's called natural competence for transformation. And what this is that if the bacterium enters the competent state, as illustrated here, it is able to take up free DNA from the environment, and in case it incorporates it into its own genome, we talk about a natural transformant. And so the question then, of course, is there any horizontal gene transfer? Does it occur in Vibrio cholerae at all? And so here I'm just showing you a slide and a scheme from, again, a study from Rita Colwell, where they isolated 23 Vibrio cholerae strains over the last 100 years, and they whole genome sequenced them. And what they found in these 23 isolates is that there were more than 73 genomic islands, and island was defined here as more than 5 consecutive genes in one stretch. And so what's shown here is the common ancestor of all these 23 strains that are shown around here, and all these boxes show the going in and out of different islands, including pathogenicity islands, or antigen-determining islands, and so on. So most labs, including mine, we're focusing on the current pandemic strains, so 7 pandemic strains that are shown here, and you can see they contain some additional islands that some of the other strains don't have, including the main virulent islands, of course. So in this study, Rita Colwell and her collaborators, they actually concluded that there seems to be extensive genetic recombination based on horizontal or lateral gene transfer in Vibrio cholerae and other Vibrios as well. So to come back to this slide, as we knew chitin induces this competence program, my lab then was interested when I started several years ago as how this is regulated, the whole competence program. And so I'm showing you that because you will understand like a bit later why this is important for my talk and what the connection to the type 6 secretion system is. So what we knew for a long time is that when Vibrio cholerae grows on chitin, it uses a main regulator, that's a transformation regulator called T-FOX. And then T-FOX is essential and sufficient to induce a pair, like a few genes, they're called pill genes, that make up the main part of the DNA uptake machinery. And that's shown here, and they actually encode a bona fide type 4 pilus. And Patrick Seiss, the former graduate student in my lab, could actually visualize this competence-induced type 4 pilus by immunofluorescence against the major pilline subunit, which is shown here. However, this DNA uptake machinery pilus is not sufficient to pull DNA into the cells, really. And again, we knew for a while that there's also a secondary pathway that's required to induce full competence in Vibrio cholerae. And that's based on quorum sensing. And I guess you are all familiar with quorum sensing, but it's just to briefly mention, it's a way for bacteria to communicate with each other or to measure diffusion, maybe, as we heard this morning, based on the production and sensing of autoinducers. And while quorum sensing has been extensively studied by Bonnie Basler, mostly, and her collaborators, and there's a huge network in between the high cell density state and the production of the major regulator of quorum sensing HAB-R, I'm not going to address any part in between here, but you can imagine this is not trivial, this regulation here. So for us, it was important that once the bacteria at a high cell density state, they produce this regulator HAB-R. And Mirella Loscutato, again, a former graduate student in my lab, showed that together T-FOX and HAB-R then lead to the production of the quorum sensing and T-FOX dependent regulator QSTR shown here, which then itself is important for the activation of a few other competent genes that are part of the DNA uptake machinery. And so one encodes COMI-A, which was predicted to be a periplasmic protein, and the other one COMI-C, which is most likely the inner membrane transporter to shuffle the DNA into the cell. So the question we had is, how does this machinery then work in pulling DNA inside the cell? And so here I'm showing you, again, a study from Patrick in my lab. So what we investigated is the COMI-A protein in the periplasm. And you see here a fully functional fusion of COMI-A with Chaire. And what we observed is that the COMI-A protein is really localized evenly in the periplasm. So it doesn't have a specific location around the cell, but it's evenly spread in the periplasm. However, this changes once we add DNA, because the bacteria are competent and they start to take up this DNA. And so what you will see in this very short movie is that then the protein relocalizes. And so these are 3-second images, and it's a very short movie. And we observed that very efficiently this protein forms one discrete foci where the protein accumulates. And so based on thousands of image analysis, we realized it's always happening in this manner. And what's happening there is really that the pilus most likely retracts and thereby open up the pores, the secretion in the outer membrane, so that some of this DNA can stick into the periplasm. Now, the COMI-A protein, if purified, has an extremely high affinity for double-stranded DNA, and it acts as a Brownian ratchet and thereby pulls in the DNA into the periplasm. And that's why we always see this COMI-A aggregate or accumulate where the DNA comes inside, and we have also studies where we co-localize the incoming DNA with this protein. So this is important that you understand later on what I'm going to tell you now. So in this study that I'm telling you, we had the question, what else is there? Is there something that's actually co-regulated with this competence regulon? And so the approach we took is that we produced this T-FOX transformation regulators at very low abundancy, either in a wild-type strain or in a strain that lacks the quorum sensing regulator HAB-R or the quorum sensing and T-FOX dependent regulator QSTR. And so we then used an RNA-seq approach to understand what might be co-regulated with the DNA uptake machinery. And to our surprise, what we observed is that the type 6 secretion gene cluster, which is shown here, that's the major gene cluster that encodes for most of the structure component of the type 6 secretion system, and I will tell you in a minute what the type 6 secretion system is, was actually upregulated when we compared a wild-type Vibrio cholerae strain without T-FOX induction or with T-FOX induction. And most of these genes were between 5 and 15-fold upregulated upon production of the T-FOX protein. Importantly, though, when we used a strain that lacks the main quorum sensing regulator HAB-R or the quorum sensing and T-FOX dependent regulator QSTR, even upon T-Fox induction, there is no upregulation of this gene, indicating that it's exactly co-regulated with the part of our DNA uptake machinery that's important for pulling DNA into the cell. Now, when we get this data, and we were wondering, is that a common theme to pandemic strains? Because many labs work with different isolates, and the isolate we have, and many other people are also working on this isolate, is from South America. So we checked this in a plethora of other pandemic strains, and I'm showing you some of these data just here. So here we did qRT-PCR, and on the y-axis you see the relative expression. And we always compared a wild-type strain without and with T-Fox induction, and in the same strain background, HABA negative strain without and with T-Fox induction. And when we look at this competence protein or gene here, we see that upon T-Fox induction is highly upregulated and fully dependent on HABA. And then these 4 genes that are representative of this large type 6 secretion system cluster that I just showed you before, we see the same phenotype or the same upregulation of these genes in a T-Fox dependent manner and fully dependent on HABA. So this was an isolate from Bangladesh from the 70s. We used a commonly used pandemic strain from Bahrain. We see the same picture. We have a very recent patient isolate that we got from our collaborators in the Democratic Republic Congo, and the same for the first sequence strain of Vibrio cholerae with N16961, which we repaired for its HABA frame shift as it's a quorum sensing mutant initially. So it seems this is a common response if T-Fox is on, that the type 6 secretion is upregulated. Now, what is the type 6 secretion system? And so I'm showing you a low resolution structure here, and then Eric, who talks after me, will basically show you beautiful structures of how the system really looks like. So what we have here is something that looks like an inverted phage tail that's attached to the inner and outer membrane. And we have here the membrane spanning complex and base plate, and inside here is an inner tube that you can't see here. And the tube is decorated on the outside with 2 sheath proteins that are called Vib A and Vib B. Now, if the system is contracting, or the sheath is contracting, it propels out the inner tube that's made of HCP outside of the cell, and it can then target neighboring cells. And if there is a neighboring cell, we call it the prey, compared to the attacking cell, the predator. And this, what I just summarized here in a few animation, was really enormous work that has been done by many groups, and some of them are indicated below here. John Michelanos and his group, it was initially discovered that this is a secretion system, and then also Joseph Mujuz and Eric and Stefan Pokatsky and many others that you see the names here. Now, for Vibrio cholerae, it was known that what the pandemic strains deliver with this type 6 secretion system, and mostly with toxins that are attached to the tip, are different toxic effectors. And the first one is a cell wall degrading or peptidoglycan degrading enzyme, so it chops up the peptidoglycan layer. There's a lipase, and there's a pore-forming toxin. Now, what's important for type 6 secretion is that the producer doesn't kill itself or its siblings, so there's kin discrimination because they produce the immunity proteins against these toxins, so they don't kill themselves or their siblings. And despite the fact that we knew so much about the type 6 secretion system in Vibrio cholerae from John Michelanos' work and his former postdocs, most of the studies on the structures were done in non-pandemic strains, non-pandemic strains, because the system is constitutively activated in these strains, so it's very convenient to basically look at the function of the system, because the system is always on. And one specific strain, and that's where Stefan Pokatsky actually identified the type 6 secretion system initially, is strain V52, a non-pandemic strain where the system is always activated, and I will come back to this strain later. So, but that also tells us that we were not really sure about what the environmental trigger for the type 6 secretion system is for these pandemic strains. And that also tells us that we missed something about the biological function of the system. So based on the RNA-seq data that I just showed you, we basically concluded that T-Fox, the transformation regulator, is not only inducing the DNA uptake machinery, which we knew for a while, but in addition is also activating the type 6 secretion system. And so for now, we've only done that by inducing T-Fox at low levels. And the question was, is this really what's happening when the bacteria grow on these chitin surfaces in the environment? So the approach we took was, again, a microscopy approach, and we used a translational fusion between the sheath protein Vib-A fused to superfolder GFP, and that has been described before by Marek Basler, while he was still in John Michelanis' lab, and the only difference that we used here is that we exchange it for the indigenous gene so that it's at the native locus and also regulated in the native way and not titrated it in trans. And at the same time, we also looked at a COMI-A protein, again, that I introduced to you in the beginning, as it's important for later on. So when we look at chitin colonization and gene expression on chitin, we use so-called chitin beads. And here you see a phase contrast image of how this looks like, because we can put it under the light microscope. And the bacteria then attach to these chitin surface and colonize the surface, and they use this as the sole carbon source. There is no other carbon source in this growth condition. And then we used, we had the strain that has the two translation fusion, and if we zoom in, what we actually saw is that indeed all the competent bacteria that have this COMI-A protein, that's important part of the DNA uptake machinery, that they also produce the type 6 secretion protein, sheath protein, Vib-A. And importantly also, and that's indicated by the little arrows here, they also make this very elongated structures, which is an indication that the type 6 secretion system is really assembled on these chitin surfaces. Then we can do some short time lapses on this chitin surface. It's a bit challenging to do these. So here you see just the 10 minutes time lapse, take an image every 5 minutes. And we see that these long structures actually contract within 5 minutes. And here is a strain which has 3 type 6 secretion systems. And then 5 minutes later, one is already contracted and completely disassembled. So the system seems to be induced on the chitin surface without any artificial induction, and it seems to be assembled and active. Now, the type 6 secretion system, it's a killing device. It's really a molecular weapon because it injects something into neighboring bacteria. So we were wondering, is that what is happening here if we induce competence and the type 6 secretion system, do the bacteria kill other bacteria? And we used a standard killing assay where we put Vibrio cholerae together with a prey, and we used E. coli in this assay. So if we look at the recovery of the E. coli population, and this is shown here on this agar plate at 10-fold dilutions, what we observed that if we put it together with a wild-type Vibrio cholerae strain, in the absence of T-Fox induction, we get really good recovery of the E. coli prey, but there's significant killing once T-Fox and competence is induced. And this is fully dependent on the type 6 secretion system, as a mutant that lacks the system does not show this killing behavior anymore. And this can be quantified, and what we see here is that through the really low level induction of T-Fox, we see 2 to 3 orders of magnitude, or 100 to 1,000-fold reduction of the E. coli population in this assay, and this is type 6 dependent. Now, if we go back to the initial question, Vibrio cholerae in the environment, obviously it's very low chances that Vibrio cholerae and E. coli would meet. So we wondered what is really happening out there in nature. And so we took the following approach, and we used 2 Vibrio cholerae strains and put them together. And so the first strain is an environmental isolate from California. So it's a non-pandemic strain, and we labeled it in green, so it's GFP positive. And we put it together with our O1L2 pandemic strain, which is the predator, and you will recognize it by the red halo again, because that's an indication that competence is on. Now, what's important, even though these are 2 Vibrio cholerae strains, they have different type 6 secretion toxins. So the 3 toxins that I mentioned in the beginning, the peptidoglycan degrading toxin, the lipase, and the pore-forming toxin, does not exist in the environmental strain. It has different toxins, and accordingly, it also has different immunity proteins. So there's no kin discrimination because these are different toxins and immunity. What I should also mention, in this specific experiment, we also knocked out the type 6 secretion system of the prey because we wanted to see a one-way fight, so to say. Now, if we put them together and we induce competence in the predator, we observed by looking at the prey that it indeed rounds up, which is a clear indication that there's a problem with the cell wall, with the peptidoglycan layer. And if we look in the phase contrast image, we also see that the bacteria lose their phase contrast, and we see also some ghost cells that already lost the fluorescence, so we cannot see them in the green channel anymore. And at the moment, we're also setting up scanning EM to really visualize what's happening there on chitin surfaces by putting these strains together. And I think you can appreciate that these don't look very healthy anymore, and they really lose their shape and they completely lyse. What was very surprising for us, though, once we looked at the predator, because what we see now, the predator is clear red, so the competence protein COMA is produced. But what we also observed is that we already get, again, all these accumulated COMA proteins. And I remind you what I told you basically at the beginning of my talk is that this is a clear indication that the bacteria have taken up DNA because the COMA protein then binds to the DNA and pulls it inside. So to show you some more examples, so here's a view where we see the prey and the predator together, and we see that the prey really lyses when we put it with a wild-type predator, and we see all this foci because the predator has taken up DNA. If we do the same experiment in a predator that has the type 6 secretion system deleted, we don't see the same picture. So the prey are healthy, they look Vibrio shaped, they are not rounding up because they are not attacked anymore. And as a consequence, we also don't see accumulation of the COMA protein because there's no DNA uptake. And as a last control, what we basically did is we put the wild-type predator together with the prey, so the prey rounds up and lyses as expected. But at the same time, we put extracellular nuclease to degrade any prey-released DNA. And in this case, again, we don't see this accumulation of the COMA protein because there is no DNA available in the environment. And so here I'm just showing you two quick movies. They are actually 6 hours movie, and I'm showing you 2 hours, but it's obviously fast-forwarded. And so in this movie, I want you to focus on the cell. The prey is already rounded up, and the predator is next to it. And what you saw here, and I'm showing you a second example here, is as soon as the prey lyses, that we get very efficient and quick uptake of the DNA by the predator. So here's another example. So the predator here in red, it also actually has the type 6 secretion system as these green spots. And so what you will see is this is the prey, this and this is going to lyse. And then right away you see that the predator is taking up the DNA and we get this accumulation of the COMA protein. So it's within very few minutes that the DNA is directly transferred. However, based on these images, we could only conclude that the DNA is taken up in the periplasm, and that per definition is not a horizontal gene transfer because it doesn't change anything in the genome. So we were wondering if the effect of this DNA transfer is really that DNA is taken up in the cytoplasm of the bacterium and recombined into the chromosome. So to test this, we did the following experiment. We put a prey that was labeled with the antibiotic resistance cassette together with the predator that was resistant against another antibiotic, and we basically tested the transfer of the cassette. And so what you see here on the y-axis is the transformation frequency. And if we use a wild-type Vibrio cholerae strain without T-Fox induction, we're below detection limit. There's no transformation that's occurred. However, if T-Fox is induced, we get the induction of the type 6 secretion system, so the lysis of the prey, and at the same time, the prey-released DNA is taken up, and we get between 1 in 10 to 3 and 1 in 10 to 4 transformants based on the transfer of this resistance cassette. This is fully dependent on the COMA protein. As I told you, it's required to pull in the DNA into the cell, but also what we observed is that in the absence of the type 6 secretion base plate and membrane complex, we don't see any transformants, and in the absence of the sheath protein of the type 6, we see a very significant reduction or even below detection limit of transformant, indicating that the type 6 secretion system really contributes to make prey DNA available. And so this brings me to the summary of my first part. So what's happening out there is that Vibrio cholerae has really high affinity for chitin surfaces, and it's been known for a long time from Saul Roseman's lab and others that Vibrio cholerae, and actually a lot of other Vibrios as well, have high affinity and chemotax towards chitin surfaces. Because as I told you in the beginning, this is the carbon source and the polysaccharide in the aquatic environment, and that's why a lot of bacteria are chitinolytic. Now they grow on these chitin surfaces and they form biofilms, and that leads to the high cell density state here. And as I told you, we knew from the regulation of the system that the chitin signal together with the high cell density state induces the competence program, which is shown here in this box. Now, in this study, what we've shown actually is that the type 6 secretion system is part of the competence program, and it is induced under this chitin and high cell density conditions and lead to the killing of neighboring bacteria that are next to Vibrio and are non-immune. Now, the bacteria, the prey, then lyses and releases its DNA, and as the type 6 secretion system is co-regulated with DNA uptake machinery, the predator can right away basically take the DNA into the cell, and in case it acquires new genetic material, it leads to its evolution, which is shown by this green stripe here. So what we basically argue is that the type 6 secretion system is used in the system to kill for DNA, so that the bacteria, really the aim of this induction of the type 6 was at the same time and completely co-regulated with the DNA uptake machinery is to acquire new DNA. And so if we look at that, that we have this forced killing of neighboring bacterium coupled to the competent bacteria that take up the DNA, then we wondered, actually, maybe this is a very common theme, because there is a recent study by Jan Willem Wiening, and he might talk about it afterwards, and also from the David's lab in the US, that showed that actually there's also a coordinated kin-discriminated neighbor predation coupled to competence in Streptococcus pneumonia. So it might be a common theme that competent bacteria make the pool of available DNA increase by predation in a kin-discriminated fashion. So for my second and shorter part, I just want to come back to the regulator T-Fox. And so it's a quite unusual regulator, and to tell you the truth, we don't really know how it works. But we knew for a long time that it's regulated and induced specifically when Vibrio cholerae grows on chitin. Now it turns out it has two domains, and as we don't know how it works, these are annotated as T-Fox N-terminal and T-Fox C-terminal domain, and there's no homology to other domains. If we look in the databases, there's more than 3,800 proteins, bacterial proteins, that have these two domains, and even many proteins that have either of the two domains. And these are actually spread here for the N-terminal domain and the C-terminal among all gram-positive and gram-negative bacteria. So we have the Vibrios here, but we also have other gamma proteobacteria. We have Firmicutes, Clostridia, Streptococci here that have the N-terminal domain. And here for the C-terminal, so it's a very abundant protein domain. And so what turns out that whenever this domain is seen in a protein, it's annotated as a competence regulator, because that's what T-Fox does. However, there's a homologue of T-Fox, which is called T4Y, that exists in Vibrio cholerae. It has these two domains, it's the same two domain structures, it's 35% identical. And it turns out, and that has been shown more than 10 years ago, and again by Ned Ruby's lab, also for Vibrio fischeri, that all Vibrios have actually two homologues of T-Fox, so T-Fox and T4Y. And again, T4Y, as it's a homologue of T-Fox, it was annotated as a competence regulator. Now it turns out when we looked at it, and we looked at a transformation we've shown on the y-axis in a wild-type Vibrio cholerae strain and compared it to a T-Fox null, where there's no transformation at all, in the absence of T4Y, it doesn't change at all. So T4Y has zero role in natural competence and transformation in Vibrio cholerae. So the question that we then ask is, so what's it doing? So what's the signal that induces T-Fox? And more importantly also, what's it regulating? What's downstream of T4Y? And so we used again an RNA-seq approach, And we were really surprised when we saw that T4Y is also regulating the type 6 secretion system. So here I'm showing you again a microscopy image. So if we induce T4Y at very low levels, and we have again the translation and fusion of the type 6 sheath protein fused to superfolded GFP, we see that they make really beautiful structures and that the system is regulated by this regulator. So that seems that these two regulators have in common, and they both regulate the type 6 secretion system. However, when we looked here again at the T4Y-mediated type 6 induction, was shown here, compared to T-Fox, where the signal is always higher, I have to say, in the control. When we looked in a quorum sensing deficient strain, so it lacks the main regulator of HAP-R, of quorum sensing HAP-R that I introduced to you before in the regulatory circuit, we actually see that while T-Fox requires HAP-R as co-regulator, T4Y works completely independently of quorum sensing. So it's a second regulator of type 6 secretion system, but it works very differently than the first regulator that we've identified. Now, I told you that there has been a lot of studies on the structure of the type 6 secretion system in Vibrio cholerae, and that was mostly based on a strain. It's a non-pandemic strain. It's called V52. Now, if we look at what V52 does when we put it together with E. coli in a killing assay, and here again is showing the surviving E. coli on the y-axis, if we usually put the pandemic strains as they are silent in the lab without the inducer chitin, we don't see killing, but this V52 strain, as it has this constitutive type 6 secretion system on, we see that it very efficiently kills the E. coli prey. Now, what was interesting for us when we now delete the second regulator, T4Y, in this constitutively active strain, we almost abolish the type 6 secretion activity, so it doesn't kill the prey anymore. And this can be partially complemented by providing a second copy or a real copy of T4Y in trans. So it seems that this strain is really constitutively active for its type 6 secretion system because of this regulator T4Y. So basically, we were wondering then what does T4Y does apart from regulating the type 6 secretion system. So we knew it's not required for competence. But based on the RNA-seq data, we saw that in addition to the type 6 secretion system, it also induces other toxins. And so the first toxin is a lecithinase. So it's a phospholipase C enzyme that basically degrades phospholipids or cleaves them. And indeed, when we look at a lecithinase assay, so it's an egg yolk plate where we compare a wild-type Vibrio cholerae, and you see this precipitate around the bacterium that basically shows the activity of the enzyme. And we see that a T4Y mutant has very severely reduced activity for this lecithinase, which is considered a minor virulence factor in Vibrio cholerae. And here you see the knockout that lacks full activity. In addition, T4Y also activates yet another toxin, which is a hemolysin. And here again, if we look at a hemolysin activity on blood agar plates, we see that a wild type, which you see here, this clearing zone, so it efficiently lyses blood cells, whereas a T4Y mutant is very severely impaired for this hemolysis, and the knockout is completely deficient for hemolysis. What we also observed is that T4Y is also acting as a repressor indirectly or directly, and we don't know that yet, and for an adhesion that's required for Vibrio cholerae to attach to different surfaces. And lastly, what we saw is that if we induce T4Y at low levels, we see a very significant increase in motility. So what you see here is swarming agar plates, so low agar LB plates, and this is the control strain. If we induce T4Y at low levels, we see a very significant motility phenotype, and this is not the case for the other regulator. So what we suggested, based on the fact that this T4Y regulator induces the type 6 secretion system, and there we also saw that there's an emphasis on the toxins that are specific for eukaryotes, or that contribute to the killing of eukaryotes, and we've shown that this is required to kill amoebae. And T4Y induces these toxins much more than the peptidoglycan specific one together with this lecithinase toxin and the hemolysin, and at the same time it detaches from surfaces and swims away. So we suggest that it's a defensive escape response that Vibrio cholerae might use to protect itself against eukaryotic predators. And so this brings me to the summary and why I titled my talk, What the Regulatory Network Tells Us About Type 6 Secretion System in Vibrio cholerae. So I hope I could convince you today that the single type 6 secretion system that Vibrio cholerae has, and it's encoded in 3 or maybe 4 clusters on the 2 chromosomes of Vibrio cholerae, is regulated by 2 major regulators. The first one is T-Fox, which is induced specifically in the natural environment when it grows on chitin surfaces, which is the natural habitat of the pathogen. And together with a high cell density state and the QSTR, quorum sensing dependent regulator, this regulator leads to the DNA uptake machinery production concomitantly with the type 6 secretion system. So the result of this co-regulation is that we have killing of prey, DNA acquisition by the predator, and horizontal gene transfer. At the same time, Vibrio cholerae uses yet the same type 6 secretion system under different conditions, and even though we don't know exactly what the inducer of T4Y is, we've shown that low cyclic di-GMP concentrations certainly contribute to the production of this protein. And then T4Y also induces the type 6 secretion system, but together with hemolysin and lecithinase, so 2 non-type 6 dependent toxins, and at the same time the bacteria basically detach from surfaces and swim away because of the increased motility. So we think that this regulator is a measure of danger, as has been suggested, for example, by Joseph Mojus for Pseudomonas danger signaling, and that allows Vibrio cholerae to basically shoot out all its toxin it can before it just swims away to protect itself from predation from eukaryotic predators. So with this, I want to stop here and just thank the people that contributed to this work. So 2 former members, Mirella Loscodato and Patrick Seiss, they were former graduate students in my lab. Mirella really worked nicely out the whole regulation on natural competence, and Patrick Seiss worked on the DNA uptake machinery. And then Sandrine Bourgeot worked on the type 6 secretion system and how it's coupled with the competence and DNA uptake machinery. And then she was supported by Tiziana Schigari. And then Lisa Metzger, our current postdoc in the lab, works on T-Fox and T4Y, and got some help there from Noemi Matte in the lab. And then I also like to thank some of our collaborators at EPFL that contributed to some studies on the activity and the rearing of DNA into the cells on the COMEA protein, but I didn't have time to talk about it today. And then, of course, our funding agency, and I thank you for your attention, and I'm happy to answer some questions. Thank you.